Great. Today we change gears a little bit in terms of Alzheimer's disease. So we will talk the first half about lipoprotein receptors and its role in Alzheimer's disease, and in the second half of a little bit more about physiological role of lipoprotein receptors, which then comes back again to Alzheimer's disease. Well, I have to excuse myself. I really don't know anything else in biology than Alzheimer's disease. So everything I pick up left and right, I always refer to Alzheimer's disease somehow. I don't know. So, as you know, of course, we mentioned this now a couple of times. We have these amyloid plaques derived from the beta peptide. And we said we have the driving factor basically starting with the APP gene localized on chromosome 21. And all people, most people who have trisomy 21 will develop Alzheimer's early in life, starting around 40 years of age, due to the fact that they just produce more beta peptide since they have an additional substrate for the enzymes. And the enzymes were in regard of the generation of the beta peptide, in general, beta secretase, which is the overall term of the secretases. We have base 1 as the main secretase, and hopefully meprin beta as a second one, which will be acknowledged in the future. And then we of course have gamma secretase, and then we have the liberation of the beta peptide. Okay. Then we had on this list here other genes. We had the ApoE gene on that list, which was the best genetic marker for in risk to develop Alzheimer's disease. So I mentioned in the very first lecture that ApoE is a gene family of 3 members. We have ApoE2, ApoE3, and ApoE4. And the difference between all of them is one single amino acid. Okay, that's the only difference. We know that ApoE is involved in lipid particle transport. LDL, VLDL, HDL have all some kind of ApoE molecules in there, and therefore we know that the cholesterol level in our bloodstream plays an important role not only in heart disease, but maybe in Alzheimer's disease as well. So this was known for the longest time. Alan Roses has shown that ApoE4, when you're homozygous for ApoE4, your risk to develop Alzheimer's disease increases dramatically. So this one here in green are people who don't carry ApoE4 alleles. This one here is heterozygous, has only one ApoE4 allele, and these here are homozygous for ApoE4. So the risk increases dramatically. It doesn't mean that you will develop Alzheimer's disease, but your risk increases by 70%. So when I finished my PhD in Germany, I already worked on APP, or tried to work on the physiological function of APP. Was more or less successful, but anyway, I decided to stay in the Alzheimer's field and was looking for postdoc positions abroad. And I came across the lab of Edward Koo in San Diego, and he was kind enough to accept me as a postdoc, so I went there. And he gave me a couple of projects to think about whether I was interested in working. And one project was basically based on the idea, what is the role of ApoE4 in Alzheimer's disease? And if you think about it, I mean, when I started 10 years ago, it was already clear, well, 10 years before I started, it was already clear that this had something to do with Alzheimer's disease, and many people were already working on that problem, and started directly to work on ApoE. And working with ApoE was for the longest time very difficult, since people tried to purify proteins, ApoE proteins. You can easily generate them. Transfect bacteria with cDNA, and bacterium will generate ApoE for you, no problem at all. The problem with that is not physiological ApoE. ApoE in your body is always embedded in lipids. So you need lipidated ApoE. So when you go out there now and look for the literature that is out there for Alzheimer's and ApoE, you have tons of literature working with non-lipidated ApoE and tons of literature working with different kinds of lipidated ApoE. And the results are all over the place. It's really hard to pinpoint what is right and what is wrong, right? So therefore, we decided then not to focus on ApoE to solve the role. Weird approach, I agree, but one can think of it in the way that when you think of ApoE as a ligand, which ApoE basically is, it has to be maybe the receptors for ApoE play a role in Alzheimer's disease. And one receptor that we focused on was the so-called low-density lipoprotein receptor-related protein. I'll introduce to you this protein in a minute. But this receptor binds numerous ligands, a lot of ligands. Lipases, for example, or TPA, which is a molecule, it's an enzyme which is being clinically used these days to treat stroke, alpha-2-microglobulin, or ApoE. So we thought, okay, maybe this is an interesting receptor to work on. Maybe this receptor has anything to do with Alzheimer's disease. And at the same time, a number of publications came out which pointed to a role, a genetic role of LRP1 as a marker gene for Alzheimer's disease. Well, I think this turned out not to be true. So there are a lot of correlative studies that have shown LRP1 is somehow related to Alzheimer's disease, but I think there are many more publications which have clearly shown that it's not. So a genetic marker, we can pretty much rule that out. But anyway, those days I had no clue. Nobody had. So we started to work on that because we thought maybe it has a relation to Alzheimer's disease. But to be fair, when I now say LRP is not a genetic marker for Alzheimer's disease, does that mean it has nothing to do with Alzheimer's disease? What would you say? Do you need a genetic correlation that a protein has something to do with Alzheimer's disease afterwards? No way. No way. Here, APP wild type. No correlation to Alzheimer's disease genetically. Base 1, no correlation to Alzheimer's disease. Yeah? Presenilin, wild type, no correlation to Alzheimer's disease. I mean, these proteins have a correlation when they have a mutation. We have familiar Alzheimer's disease. Okay, but sporadic, nothing. Okay, so except the only real exception is ApoE. I mean, that is solid genetic marker for Alzheimer's disease, for sporadic Alzheimer's. So all these down here turn out to be not true, or maybe true, maybe for alpha-2-microglobulin. But I'm not a geneticist. I really don't know anything about analyzing those kind of data and how strong and how hard these data are. But alpha-2-microglobulin is interesting because it is the ligand for LRP1. So many years before it was termed LRP1, this was termed alpha-2-microglobulin receptor. So here, then I would say maybe again some link to Alzheimer's disease. Good. Let's focus on LRP1. LRP1, the low-density lipoprotein receptor-related protein. I won't say that all the time, so I say LRP1 is a member. Is a member here of the LDL receptor family. And the LDL receptor family is depicted here, and of course the name sake was the LDL receptor. And all the other receptors somehow are built up in a similar shape with more retentions, so that you have a very huge receptor here for LRP1. This one is around 600 kilodaltons in size. It's a huge protein. And nobody would have cared about these receptors maybe if one hasn't started to make knockout antibodies. So at the very beginning, when the knockout technology was available, people knocked out the LDL receptor in mice. And if you think about it, you knock out the LDL receptor in mice, these mice should die right away, because they shouldn't be able to take up any cholesterol whatsoever. But interestingly, these mice live happily ever after. They couldn't care less. And that was then when people got aware of additional receptors that could take over the function of the LDL receptor. So by that, then, researchers realized, okay, there are other receptors, and all the other receptors were cloned and identified shortly afterwards. So LRP1 is one of the biggest receptors in humans anyway, 600 kilodaltons in size. It is a kind of a scavenger receptor for lipoproteins. It is expressed in neurons as well. It is, as APP, a type 1 transmembrane protein, meaning you have the N terminus extracellular, C terminus intracellular. It's cleaved here by the enzyme furin, and that happens, LRP is generated as a single-strand molecule in the ER. It is 600 kilodaltons in size. Then in the Golgi compartment, furin cleaves LRP here, and the alpha chain, or heavy chain, stays non-covalently attached to the beta chain, to the light chain. And both of them stick together as a receptor and travel to the cell surface. And there they bind then extracellular ligands, and this receptor is ubiquitously expressed with the highest expression in the liver, but high expression in brain as well. Okay, we have 4 different binding domains so far identified at LRP1, and these binding domains altogether bind maybe 70 ligands or maybe even more, I don't know, but a lot of ligands. The problem with this receptor is when you start working with such a receptor, or when you're a naive PhD student on postdoc like I was, you always think, oh, no problem, I'll take this one, I'll clone it, and then I'll transfect cells and the cell will express that. Well, good luck with cloning something like that. I couldn't do it, but luckily somebody else could do it. Sent me the construct, and then tried to express a plasmid with a 13,000 base pair construct in there. Yeah, forget it. I mean, yeah, you can do that, but you have very little expression in the cells. So you have to get around that and try to generate smaller constructs that mimic the function of this protein. Okay, so biosynthesis of LRP1, it's generated as a full-length protein in the ER, travels through the ER, and uses a specific chaperone, which is known to bind all lipoprotein receptors. And this will be important during the talk, because lipoprotein receptors have a specific chaperone just working for themselves, helping in folding the protein so that such a huge protein can get into the correct shape. So together with RAP, the protein travels to the cis-Golgi compartment. There, RAP is released, travels back to the ER, and full-length LRP travels to the trans-Golgi, where it's cleaved by furin. The both light chain and heavy chain stick together, travel to the cell surface, and here LRP binds its ligands and produces, after its single transduction reactions, or is internalized into the endosomal lysosome compartment. And there the ligands are degraded, and LRP is recycled to the cell surface. LRP is one of the fastest receptors recycled in the cell. It's very fast kinetics, and that will play a role in a minute as well. So when I first started to work with LRP, we generated an antibody, it was called 1704, and that antibody was directed against the last 15 amino acids of LRP. So when we do a Western blot with such an antibody, what you will see is full-length LRP. So this is LRP that is still in the ER, not cleaved. You have light chain LRP, so this one here. This is the LRP that is cleaved with after furin cleavage. Then you lose the heavy chain, since this is a denaturing gel. All the non-covalently attached parts will fall apart. So you detect only light chain, beta chain here, and you detect this here, bands down here, which are something like this, CTF, C-terminal fragments. You've heard that already for APP. So all the PhD students here. If you ever work with Western peoples, there's one thing you are never allowed to do, which I did, okay? Never ever cleave or cut your blots apart. So what I always did was, when I did these Western blots, I cut my blot here and was using the lower part to develop it for actin or tubulin to have a loading control, right? And then one day I forgot to cut my blots. I was working 2 years on the project and developed the whole thing. And this here popped up. And since I'm in the Alzheimer's business, I knew this one is a C-terminal fragment. What happens with C-terminal fragments of APP? They're cleaved by gamma-secretase. There. We used presenilin knockout cells, which we rescued with presenilin, and there the C-terminal fragment is gone. So presenilin does not only cleave APP, but cleaves LRP as well. Well, we realized that, wrote the paper, were ready to submit, and then a paper by Joachim Hayes came out showing exactly that. And we could have, I was just plain stupid, right? I mean, if I wouldn't have cut my blots all the time, I had a nice paper those days, but this was then useless. Okay, so never, never, ever cut your blots. So when PhD students now come to me and they cut their blots apart, I'll just send them back. Okay, so LRP is a substrate for gamma-secretase as well, like APP. LRP is, the LDL receptors are very well known through the work of Goldstein and Brown. They work in Dallas at the moment, and they received the Nobel Prize for deciphering the NPXY domain. So aspirin, protein, any amino acid entirely. And these signals in transmembrane proteins are signals for endocytosis. Whenever you see such a motif in a protein, you can be sure in the C-terminal, so the intracellular part, you can be sure this protein will be endocytosed through this clathrin-mediated mechanism, which goes back to this amino acid sequence. And LRP has two different NPXY domains in the C-terminus of the protein. So when I started then working on LRP, I started very simple and thought, okay, maybe LRP has something to do with Alzheimer's disease since it is the receptor for ApoE. So we got LRP expressing cells and LRP knockout cells. And the first thing I did was I just made a Western blot to detect APP in these cells. So LRP expressing cells, LRP knockout cells, and in both of them is APP. But in the knockout cells, there are no C-terminal fragments whatsoever. That I was curious, I had no clue what that meant. And then I measured the amount of soluble APPS in LRP expressing cells and knockout cells and realized, whoops, there is 2 times, 3 times more soluble APPS alpha in cells that do not express LRP. So when you now think of this, what we have depicted yesterday, we said we have the A beta sequence, we have the plasma membrane, and usually we have beta cleavage and gamma cleavage. And we have here in between alpha cleavage. So when we have an increase, right, in APPS alpha, which is this part here, if that goes up, you have less substrate for beta and gamma. Therefore, if you have no LRP, you shouldn't generate a beta peptides. So that would mean LRP in your body is bad, theoretically, right? The problem is you cannot, well, if LRP is really bad, then the easiest thing to do is knock it out and see what happens, right? People knock LRP out in mice, and what happens? The mice die. They die at E10 or 11, so in the embryonic phase very early. So it can't be that bad. So we certainly need it. It has a lot of functions. But unfortunately, that seems to be involved in APP processing under normal conditions, meaning this is all endogenous LRP. There's no overexpression or anything, it's just knocked out. So APPS goes up 2 times. Then I tried to analyze what is going on with the internalization rate of APP in LRP knockout or expressing cells. And you can see here, regardless which APP we use, whether we use the shorter form or the longer form of APP, LRP knockout cells internalize much less APP from the cell surface. And only when APP is internalized into the endosomal compartment, most of the A-beta is generated. Therefore, we try to rescue this in one experiment. So rescuing means when you have a knockout, you bring back the actual gene, so LRP back, and then the normal phenotype should be restored. We did that here. We brought back a part of LRP and APPS secretion goes back up. So that was a clear indication LRP had an effect on the internalization of APP. So when you have less internalization of APP, you have more at the cell surface, more alpha secretase can cleave, you have more APP is alpha. Simple as that. So is that then true that we have less A-beta production? Oh yeah, we have a lot less A-beta production when you have no LRP. You have very little A-beta production because apparently LRP drags APP together into the cell in the endosomal compartment and then A-beta is generated. And again, we can rescue that when we express an expression construct, a truncated expression construct of LRP, A-beta production goes back up. So these proteins work apparently together. And this is not, I mean, it was fantastic that we found that, but when you think about it, when we always think about proteins, whether you're a cell biologist or biochemist or geneticist, whatever, we always think of proteins as single molecules in a cell. We always say, oh, that protein is in the R, goes to the Golgi, to the cell surface. We don't care whether 10 or 20 others are attached to them and maybe in different combinations and do specific things with it. I mean, otherwise, if that wouldn't be true, we could not explain why we exist with a little bit more than 24,000 genes. Well, that's nothing. Worms have more genes than we have, right? I mean, it can't be the genome what tells us that you are you and I'm me. It has to be genes plus the proteome, meaning the combination of proteins, different combinations of proteins that fulfill certain functions. So that means many proteins are involved in a process that seems to be so easy. APP is internalized, A-beta is generated. And then we take one other protein away and everything is messed up. So how is that then possible? That if we take away this huge protein, APP is somehow affected? First, we thought, okay, maybe the interaction between APP and LRP goes through the N-terminal part, so outside of the cell. There was a publication showing that this domain here, the so-called KPI domain, is involved in the interaction. The problem with that, to me, was KPI domain is only expressed in the long forms of APP, 775 or 770, and not in 695. And 695 is the form that is mainly expressed in neurons. So if this is then the only interaction partner in neurons, this shouldn't work. But it works in neurons. So I never thought that this could be the only driving force in it. So then we thought, okay, maybe through the C-terminus we have some kind of interaction, either directly or through different adapter proteins. So another, or more proteins come into the game. And there are different adapter proteins like DAP1, X11, or Fe65 were possible because all of them have been shown by then to bind either to LRP or to APP. Some of them were shown to bind both. So we tried to investigate that and again use our knockout cells, our LRP expressing cells, and we generated a truncated LRP construct, because now we needed a construct that pretty much lacked the N-terminus of LRP. If we want to exclude that LRP interacts with APP at the N-terminus, we just cleave off, zack, here, everything up here. And if this one, the short one, works, we know it's the C-terminus that matters, right? So we generated a very short LRP construct which still reached through the plasma membrane. And here we try to come up with an approach to show that APP binds to LRP through a third protein. And if you think of it, it took us a while when we started it in, it was, I don't know, 2000 or something, to come up with an idea how to show on a very easy biochemical level, we had no fancy machines or anything, to show how three proteins directly build a complex. So the easiest thing, I was naive, thought, okay, what I do is, I do an immunoprecipitation for LRP, and then I come back with an antibody for Fe65 and APP. And therefore, I've proven all of them are in one complex and stick together. But if you think of it, like I explained it here, so I would say I come with an antibody against LRP1, right? And then I pull down at the same time Fe65 and APP. But this doesn't mean that this one connects LRP to APP. They just stick all together in one complex. So what we had to prove is that this one is in between there. And that's difficult. So what we did was the following. We used our LRP knockout cells, which were stably transfected with the LRP construct, with the truncated LRP construct. And then we used a soluble protein which was only reflected the C-terminal part of APP, so only this part here, expressed that in bacteria together with the GST tag. So when we had that protein, we could later use this GST tag to bind that to beans. Okay? So we had this lysate, we had these cells, and we transfected these cells either with nothing, we remark, or with an Fe65 construct that lacked one binding domain, or with an Fe65 construct that was completely intact. And when we then came back with this soluble fragment here, the GST tag APP fragment, and pulled down the whole lysate, we could only see, when we came back with an LRP antibody, LRP here, because then APP was pulling down Fe65, and we could show that LRP is in there. And when we truncated this Fe65 construct so that this domain couldn't bind to APP anymore, we couldn't see anything at all. Meaning, a tripartite complex out of these 12 proteins. Then we reversed the experiment, pulled with an LRP construct, and saw APP coming down. This is on a radiolabeled basis. Same result. So those three are in a tripartite complex. Okay. So now, I hope I could have convinced you that those three proteins stick together and play a role in APP processing. So then I thought, okay, great. Then the next thing I'll do is I overexpress Fe65 in my cells. Therefore, I should get more complex formation, more LRP together with APP. And when I have more complex, I probably get more internalization of both proteins, and therefore maybe even more integral. Right? That was my idea behind it. But what happened was the following. I used cells that overexpress APP, transfected in Fe65, or the truncated form of Fe65, and measured soluble APP as alpha. So I got again an increase in this here. I measured A beta. I got almost no A beta. And I expected the complete opposite. I was stunned. I had no clue what to do. It took me a week to figure it out, and then I thought, maybe this is happening. So if this is the case, under normal conditions, that LRP binds to APP through an adaptive protein here, Fe65, and we now overexpress Fe65, maybe Fe65, we have so much Fe65 in there that one molecule binds to APP, the other one binds to LRP, and by that, we break up that complex. And when we break up that complex, we have basically the LRP knockout situation I've described to you in the very beginning. More APP is alpha, no beta. Same is true here. This one can only bind to LRP, it cannot even bind to APP. Therefore, we generated again an APP knockout, an LRP knockout, sorry. And again, same result. So if this is correct, then this is what we expect. I just didn't think of it when I overexpressed that protein. So usually when you overexpress a protein, you go in with some expectation, and whether that's at the end correct, you have to figure out for yourself. But here, clearly, this proved to me that I was wrong. But anyway, it worked nicely in the way that this was my assumption. So if this is now correct, I should be able to show that. So when I overexpress Fe65, I should get less complex, right? So this is what we did. We used the neuronal cell line, which expressed LRP here and here, and we immunoprecipitated for APP, and then came back for with an LRP antibody. So when we express in there no Fe65, or not the truncated form, you get a certain amount of LRP down, here and here. When you overexpress Fe65, the truncated version, you get less complex down. Or when you express Fe65 full length here, and you come back with an LRP antibody, you get less complex down. Okay, so that was proof that when you overexpress that protein, you break up that complex. And funny thing happened when I presented that data, or when we published that data. I went to a conference afterwards and met Luc Merken, and he was pretty happy with our paper because what they did, they had generated mice for even 3 years before I was working on that story, and what they saw in their mice was the following. These are regular APP overexpressing mice, plaques all over there. And when they transfected Fe65, they were, like me, expecting to get more A-beta. And they saw nothing. No plaques whatsoever. And when they saw our paper, and they mentioned it nicely, that, yeah, you don't produce any A-beta anymore, most likely to the effect that I have presented to you a minute ago. You see here again, this is a brain lysate. If you overexpress Fe65, A-beta is pretty much gone in the brain. Okay, so let's continue with the story of LRP and APP. Then we ask ourselves, where is this story started? Is LRP and APP interacting just at the cell surface or prior to that? So, as I said yesterday, we generated constructs that carry a KKAA tag. This is a tag that keeps your proteins in the ER. And you can see that nicely here, that these proteins are expressed in the ER. The retention constructs are really nicely expressed in the ER. If you don't retain the construct, you have some proteins in the ER, but not a whole lot of them. So these proteins do not travel to the cell surface. But when we retain the LRP construct in the ER, as shown here, this is LRP, these proteins retain at the same time APP in the ER. So endogenous APP is retained in the ER when LRP is retained in the ER. So these two proteins stick together already in the ER, travel together to the surface, and then are together internalized. So when you show that on a biochemical level here with surface bioaccumulation experiments, we have either mock transfected cells, or cells transfected with our LRP retention construct, or with our normal rescue construct for LRP. This is what happens. Under normal conditions, you have a certain amount of APP at the surface. When you retain LRP in the ER, you have much less APP at the surface. And when you have regular LRP expression, again, normal APP expression at the surface. Even more, when you have this, you even have C-terminal fragments at the surface. With the retention construct, you can't see it. So LRP retains APP already in the endoplasmic reticulum. So this was the story of how the two proteins interact and how LRP functions as a protein that generates A-beta, right? So we agree all, LRP is bad. Right? You have LRP, you produce A-beta. Okay. Switching gears in a different way. When we started to work on LRP, we pursued a different way as well. We tried to find out whether LRP is capable of binding alpha-2 macroglobulin, that was known already, and we were interested whether alpha-2 macroglobulin could bind A-beta at the same time, and then all of them together are internalized and cleared by cells. So whether that is a possibility for a cell to get rid of A-beta. And we could show that using different kinetic approaches that indeed LRP is capable of binding this complex here, and that is then internalized and A-beta is completely degraded. Not completely, but it is degraded. And we can block that process when we use this protein, so-called RAP, that was that chaperone from ER. You can generate that in bacteria and add it just to cells, and then this protein will smoothly bind to all surface lipoprotein receptors, regardless which one it is, and blocks all other ligands from binding to lipoproteins, to the receptors, sorry. So if you add RAP at the same time, you have no binding of this complex to LRP. So here is what happens. Under normal circumstances, you have a certain amount of LRP, of A-beta internalization, or this says it's a steady state level in the cells. LRP is normally expressed. When we add alpha-2 macroglobulin to this is a reduction in the A-beta level when you just add a little more alpha-2 macroglobulin to it, because alpha-2 macroglobulin binds A-beta additionally, binds it to LRP, it's internalized and degraded. And we can block that phenomenon completely when we add RAP to it, because RAP now inhibits binding of A-beta and alpha-2 macroglobulin to LRP, and you have a lot of A-beta still in the cell floating around. And this effect is completely gone in LRP knockout cells. Okay. So this now would tell us LRP is good, right? Because LRP takes up A-beta peptides. So we have the conundrum here that is LRP friend or foe? Is LRP, we have a high LRP expression with an increased A-beta production. But on the other side, if we have a high LRP expression, we have increased A-beta clearance. So which one is the better one? What would you think?
The question is incorrect. It's neither friend or foe, it's just process.
Correct, it's nature, I agree. I completely agree. And since we cannot knock out LRP or play around with the expression level of LRP, we have to accept that. Right? So for example, if you would think of an approach where you try to specifically knock down LRP during aging, for example, not from birth, but later on, and you will not only knock down LRP in your brain, but in your liver. And if you lose the capacity of your lipoproteins to be degraded in the liver, good night. Right? I mean, that's it. So this is a hard puzzle to solve. I would personally, what I like best is this approach here. That high LRP expression is good, Because we have an increase in A beta clearance, and we will, to this part, we will come back tomorrow, because LRP plays a dramatic, interesting role in the clearance of A beta peptides across the blood-brain barrier. And during aging, and specifically during Alzheimer's disease, you lose LRP expression in your brain, and therefore you lose the capacity to clear your A beta peptides. And that might be an additional role why more A beta peptides accumulate over time in your brain, and why you're getting Alzheimer's disease when you're old, and not when you're young. When you're young, you have high LRP expression, you produce a lot of A beta, doesn't matter. But you can clear a lot of A beta. When you're old, you still produce A beta, but you can't clear it anymore. And then you have a problem. Okay? So maybe this is a good part, I hope at least, and this part will be extended on tomorrow. So, in the second part of the talk, we will talk a little bit more about the physiological function of LRP and its role in neuronal signaling. A little bit I'll talk about stroke, and at the end I will come back to Alzheimer's disease. But before we're going to do that, I think we have a break, and Anja, didn't you want to make an announcement?
Yeah, Zina wanted to.
Okay. Now I'll dig into the physiological role of LRP1 a little bit more. And we stumbled over it because there was a publication which I personally thought didn't make much sense. So we thought we worked on it and wanted to see whether that is correct or not. So to give you a short background about LRP, as I mentioned a little bit about it already. In 1992, there was this complete knockout of LRP generated, and we have a very early lethality. So that is a problem. You can generate embryonic cells, fibroblasts from these animals, but really nothing else. And then, many years later, the group from Goujon generated a conditional knockout in the forebrain, and which is driven by the CAM kinase 2 promoter. And these animals live, but they have severe phenotypes, and I'll show you that in a minute. Additionally, there's another LRP mouse model available, which was very elegantly generated by Anton Röbbeck, and what he did was he made not a knockout, he made a knock-in. He made different knock-ins into the endogenous LRP gene. And he targeted the NPXY domains of LRP. And that was actually quite interesting because he made an amino acid knock-in into the first NPXY domain, and the mice died. So this domain has some severe effect on the whole protein, and we still don't know exactly what it is. Then he made a knock-in in the second NPXY domain, this one down here, and the mice were pretty much normal and fertile. Well, pretty much normal means he generated them and they lived. So there was no real investigation at that day. We started with the investigation a little later. And of course, the double knock-in mutation is embryonic lethal, pretty much as the full knockout. So single amino acid exchanges resulted in a complete knockout phenotype of these mice. And we got interested in this story in two different ways. So one way was looking at the animals that were generated by Goujon, where you have the conditional knockout in the forebrain. And these mice show, for example, spine degeneration. So here LRP is turned on through the Cree promoter. And you see you have less spines here, and that is quantified here. And they have a behavioral phenotype. They show a phenotype on the rotor rod or in open field testing. So these mice are, in general, let's say, a little bit down. Okay. these mice are, the promoter is turned on only after the mice are born and later on in life. But then still we see this behavioral phenotype in these animals. We got interested in the story because there was a paper out there showing that TPA can activate or can let's say activate the NMDA receptor. NMDA receptor, we talked about it on the very first day, is a receptor that is important for learning and memory. And TPA by itself is one of the best known ligands to LRP. But when that paper came out, what they showed was you use TPA, and this TPA should then have an effect on the NMDA receptor directly. And that was stunning to me. It might be true. It might be true, don't get me wrong. But at those days, I thought, no, that can't be correct. There has to be something else to it. And since I knew that TPA is one of the ligands to LRP, I thought, okay, wait a minute. LRP has a severe effect on spine generation, so maybe LRP has an effect on NMDA receptors. And maybe that effect could be manipulated through TPA. It was just an idea. Then a PhD student came to my lab and I said, well, here's a project, okay? And then that poor student has to start with that stupid idea that I have and run with it. One advice when you start a project as a PhD student or a postdoc, okay? So you go to your supervisor tells you, this is the project, do it, okay? Then you're eager to do it, and then you find out nothing works. So the first thing, when people come to my office, they show me their results, nothing works. Well, 90% of the time, they just made a mistake. Something was completely wrong, okay. So they do it again and again and again. But after a while, when you do it correctly and you can prove that you do the experiment correctly, you still find out it's completely bogus. Doesn't make any sense. Then you have to challenge your supervisor, right? And tell them, okay, there's something wrong. And then that is science, guys. Okay? Then you have to convince your supervisor, we have to change the project in this or that way. Okay? So don't get me wrong, don't argue with your supervisor after 2 weeks when you started your project. Okay? Argue with your supervisor a year when you're in your project, not before, maybe before. But that is science, okay? So we started with this idea that TPA cannot activate the NMDA receptor. It has to go maybe through LRP. And I'll try to show you the story behind it. First of all, TPA is an interesting enzyme because it's sold as a drug, and it's pretty much the only drug that you can use when you have a stroke. Stroke research has a problem these days. There is hardly any pharmaceutical stroke research anymore because due to the fact that all the trials they started failed. So the industry spent millions and millions and millions of dollars in trials and they all failed. So therefore they cut the trials and they cut the research. So we're stuck with this. And to be honest, this can happen to Alzheimer's disease as well. When the next trials with all the drugs we come up with fail, the companies will drop out of Alzheimer's disease research, I predict. And then we have a problem, a big problem. Okay. So what is TPA doing in stroke? Very simple. Stroke is basically nothing else as a blood clot in your brain, in your capillaries of your brain. And with that, then, it clogs the vasculature and you have parts of your brain deprived of oxygen and the neurons die. That is stroke. But if you detect that very fast, you really have to hurry to the hospital, you will be injected with TPA, your blood clot will be dissolved and your oxygen flow is normal in the brain. And you pretty much have only weak symptoms, if at all. Okay? But that only works minutes to few hours after the original stroke happened. After that, you're doomed. There's nothing else available at the moment, at least to my knowledge. Okay? So TPA we use in stroke therapy, just as an explanation. So coming back to the original idea, from those papers that stated when you treat neurons or young neurons with TPA, then this TPA should activate the NMDA receptor, and in our lab we started to measure that by downstream signaling from the NMDA receptor. We just chose a very common signaling cascade. It's the so-called ERK cascade, MAP kinase phosphorylation cascade. So what you can see here is when we add nothing to cells, to neurons, you have this amount of phosphorylation of the ERKs. When you add TPA, indeed you have downstream signaling, which was described in that paper. And if you add alpha-2 microglobulin, a completely different receptor, you have nothing. Okay? So that means TPA can do it, alpha-2 microglobulin cannot do it, although both of them should be ligands to LRP. Okay, then we said we take the NMDA receptor out of the equation. Maybe the effect here on signaling we see in neurons has nothing to do with the NMDA receptor. Therefore, we chose this time here Chinese hamster ovary cells. It's just a regular cell line. Doesn't matter, you can use any kind of cell line that doesn't have an NMDA receptor. And here, what happens is, if you add TPA, you see nothing. So, no effect whatsoever on ERK signaling. Meaning, you clearly need the NMDA receptor for it. So, by that day, when we saw these results, my PhD student was already arguing with me what kind of stupid idea I had. Then we said, okay, if this is correct, that LRP has nothing to do with it, we try the following. We should then be able to block LRP on neurons, and then TPA should still have the effect that we have seen before, right? And blocking LRP, as I said before, is quite easy. We just purify this RAP protein, add that to cells from the outside, and then RAP binds nicely to LRP and prevents any kind of ligand to bind. So TPA cannot bind anymore, and therefore, if LRP has no effect, we still should see phosphorylation of ERK. If LRP is an effect, we should see reduced phosphorylation of ERK. And this is what happens. So we add TPA to the cell, again neurons, we have activation. When we add TPA together with RAP, the blockage of LRP, the activation is gone. So I was back in business and I sent my PhD student back to work. Okay, LRP clearly has a role in it. Unfortunately, the NMDA receptor as well. So both of them have to be there to see an NMDA receptor mediated activation of downstream signaling. Okay, then we said to further prove that LRP plays a role in the NMDA receptor downstream signaling, we will knock down LRP. And that's a hard thing to do. But we tried, and we weren't really successful, but we were able to knock down LRP to this amount compared to vector control, so maybe a knockdown by, I don't know, 60%, maybe, I don't know. But this was enough to show that when we knock down LRP, TPA has a much reduced effect on the signaling of the NMDA receptor. That was good news, meaning both were needed. We needed LRP for that effect of the NMDA receptor. Then we said, okay, how about what happens when we now block in neurons the NMDA receptor itself? Do we then still have an effect of TPA? So you can block the NMDA receptor through different mechanisms. It's quite easy actually. There is the inhibitor MK801, which pretty much sits on top of the NMDA receptor, and you have no calcium influx into the neuron anymore. And when you add this inhibitor to neurons, you see there is no activation of the phosphorylation of ERGs. Same is true for RAP up here. So as I've shown you before, so when we inhibit LRP, you have no activation. When you inhibit the NMDA receptor, you have no activation. And even if you use a different inhibitor for the NMDA receptor, DLAP5, you have exactly the same effect. You have no activation of the NMDA receptor. So pretty good proof that both work in concert to mediate this effect. Then we thought, okay, this was all downstream signaling, meaning we had MAP kinase phosphorylation. And you ever work with cells and you stimulate cells with whatever you do, you will always get MAP kinase activation somehow. So we wanted to prove that real calcium influx is involved in that process. Therefore, we took neurons and had either neurons under normal conditions, treated with TPA, or treated with TPA together with that inhibitor for the NMDA receptor. And what you can see here is, under normal conditions, neurons start to fire. That's calcium influx into neurons. And when you block with MK801, blocking the NMDA receptor, there is baseline. So that was expected. But exactly the same, not as nice, but pretty much exactly the same, is true when you block with RAP. So when you block LRP, you have no calcium influx into neurons. So both receptors or both proteins play a role in calcium influx mediated through TPA. So this was all then kind of an artificial system, because although we worked all on endogenous protein levels, we tried to mimic it with inhibitors, so pharmacological intervention was it. We had to prove it genetically that we were correct. And for that, now we used the knock-in mice that we had available, and the only knock-in mice that were available to us which survived were the LRP delta NPXY2 mice, which survived. And we just tested whether these mice had anything, or whether LRP with the knock-in here had any effect on the NMDA receptor. So on the left-hand side, you see the animals with the wild-type allele treated with TPA for 30 minutes or 1 hour, and you have nice activation of the ERG pathway. And in the knock-in, you see much less activation here. And even after 1 hour, you have a little activation, but much less than here in the wild types. So somehow, this NPXY domain plays a role in the mediation of the signal of TPA through LRP to the NMDA receptor. So, and the same was true with the calcium imaging. So this was again MAP kinase activation, and here we measured again in neurons, calcium imaging, wild type, TPA, treatment, wild type neurons, a lot of calcium influx, and then the delta NPXY2 neurons showed very little calcium influx in these cells. Good, so that would mean somehow this part down here somehow interferes or regulates or plays a role in NMDA receptor signal. Then we thought, okay, maybe the LRP level or the expression level has something to do with the expression of the NMDA receptor. And we tested that in the following way, because what happens when you have a delta, if you have a knock-in the second NPXY domain of LRP, something very critical happens. You block the endocytosis signal of LRP, therefore you have more LRP at the cell surface and less internalization. So we thought, okay, maybe they are just not internalized together properly anymore. Or other proteins are not internalized properly anymore. So we tested just in brain homogenates the expression levels of LRP, NMDA receptor subunit 2B, 2A. So I have to say, since you're not familiar with NMDA receptors are receptors that exist out of 4 different subunits. NMDA receptor subunit 1 has always to be there in 2 forms, and then 2 others are there, either 2B or 2A. Right? And these are then functional NMDA receptors out of 4 different subunits, out of NR1 or NR1-2B or NR1-2A. But NR1-2B is thought to be more heavily involved in learning and memory. That's what physiologists say to me. So I'm really not working that much with these receptors. So we tested the expression levels, whether the overall expression levels in the brain were different between these mice. And they all look pretty much the same. So there we couldn't find any big difference. But then we looked on the surface expression levels of these proteins in primary neurons. And what you can see here, these are the lysates, so no big differences in the overall lysate expression of the proteins. But when you look at the LRP expression in neurons at the cell surface, as predicted, LRP at the surface is higher when LRP can't be internalized. And the NR1 receptor subunit is higher when you have a knock-in LRP. And remember, this is all endogenous protein levels, there's no overexpression. Same is true for the 2B subunit. The 2A subunit, not changed at all. So that was an indication that maybe LRP fiddles a little bit with the surface expression of an NMDA receptor complex out of receptor unit number 1 and 2B, and maybe not with them existing of 2A. This was then further proven by an uptake assay. Now we really measured internalization. We didn't measure surface, but we measured internalization. And you can nicely see that we have reduced internalization in LRP, in NR1, and NR2B, but no difference in NR2A. So in neurons, in primary neurons, something is completely messed up with the NMDA receptor subunit composition when we have a knock-in LRP. So would that then mean we have an overall problem with cell survival, for example? And we just did a very simple assay here. It's an Alzheimer blue assay, and you can do much more fancy assays to see to look for cell survival. But we couldn't see any difference in cell survival between wild type and P2 neurons. We tested whether other proteins in the synapse are somehow differently regulated, PSD95 or synaptophysin or whatsoever. We didn't see a big difference. So theoretically, a neuron is functional in short words. Then we wanted to see, okay, if we have this weird interplay, we should see interaction, right? I mean, that's what we always, I'm a biochemist, I always look for interaction. And so we wanted to test, okay, if we have interaction, we saw that huge upregulation of the NMDA receptor 1 subunit at the cell surface when we have a knock-in LRP. So maybe there's an interaction between that subunit and LRP. And we made lysates. You see nicely here the NMDA receptor 1 subunit in lysate, but we didn't see it in the IP. Then the obvious thing happens. My PhD student comes to me with an empty blot, and you say, or I say, of course, oh, do it again. You made a mistake. Right? I did it again. Empty. That was probably another mistake. Do it again. So she did it. He did it probably 10 times, and then I was convinced. Okay, there's nothing. Bummer. Okay. So really no interaction with the NMDA receptor 1 subunit. But luckily, there is an interaction of LRP1 with the NMDA receptor 2 subunit. Because in the immunoprecipitation, you see an increased interaction between LRP and the NMDA receptor 2 subunit in these delta MPXY2 neurons. Meaning, we have more LRP at the cell surface, you have more interaction with the NMDA receptor 2B subunit, and by that, most likely, this keeps the NMDA receptor 1 subunit as well at the cell surface, because they have to be always in a complex to be a functional receptor. So all these here that are in one receptor together bind apparently to LRP, and then can be immunoprecipitated here in terms of the NF2 subunit. Now you could say in a co-immunoprecipitation experiment we should be able to see NO1. I agree, but we don't. Okay, so there might be an interaction between those two proteins, and this might be either direct or through adapted proteins, like, for example, PSD95 or other proteins. So then we wanted to test whether these LRP mice with the knock-in in the NPXY2 domain have any behavioral phenotype. So how to measure behavior in mice? Some of you might know that, some might not. It's actually quite easy. What you do is, the simplest test is this here. It's the so-called Morris water maze test. What you do is, you take a mouse, you put the mouse into a water basin, and mice do not like to swim at all, but they swim very well, very well. And in that basin, somewhere is a hidden platform. So, meaning you don't have water in there, you have a milky kind of liquid in there, and the mice cannot see the ground, and somewhere in that liquid is a platform, just beneath the surface of that liquid, of that water, right? But the mouse can't see that. So you put the mouse here, the mouse swims around, finds the platform, sits on it, and you measure the time that the mouse needs to find the platform. Okay. Then you take out the mouse, clean the mouse, put it back in the cage. Next day, mouse back out, inside, swims around, finds the platform. Every day, again, again, again. After some time, the mouse remembers where the platform is. You put the mouse into the water, and the mouse swims straight to the platform. Okay? So that is a measure for remembrance in a mouse. Okay, so that's a measure for behavior in mice. You can do that in different ways, different mazes that you can come up with, how fast mice find their way, and you can measure that pretty good, actually. So when we do these tests now in our NPXY2 knock-in mice, you see that these mice find the platform on these in the water much slower than regular mice. So they are apparently dumber. Although we only have a knock-in in the LRP2 gene and not in the NMDA receptor at all. And this is even true when we do a little bit more fancy tests. The NPXY2 mice are a little bit dumber than the regular mice. We did rotarod as well. Here, the opposite is the case. I don't know exactly why, I have no explanation for that. So when you have a knock-in in LRP, and you put a mouse on a rotating wheel, usually mice fall off after some point in time. You can measure that. So you measure locomotion activity through this, right? The LRP2 mice, they're pretty good. So they stay pretty long on these wheels. And I have no explanation for that, but maybe, yeah, I have no explanation for that. And we did fear conditioning with these mice. So fear conditioning means you have a mouse, put the mouse in a cage, and then in that cage you have down here something that can give a mouse a foot shock, right? And when you give the mouse the foot shock, you have at the same time a noise, so that the mouse remembers the foot shock together with the noise. And then you can put the mouse in there and just give the noise, and the mouse freezes as it would have gotten the foot shock as well. And you can measure this in two different ways, in a context way or in a Q way, meaning there are two different ways to measure fear and how these mice can remember this, this process. And you see again that the LRP delta MPXY2 mice, they forget pretty fast that there was something going on in this cage, while the wild-type mice can remember pretty well. So something is going on with LRP1 and the NMDA receptor in terms of biochemical interaction and in terms of behavior. And this, again, is all under physiological conditions. So now, coming back one circle, same story, back to Alzheimer's disease. I could make exactly the same argument for Alzheimer's disease. Very simple, because since many years it is known that when you take A beta peptides and add it to neurons and you measure LTP, LTP is nothing else as currents that are basically generated through the NMDA receptor. Right? You add now, you can measure these currents, and this up here is a normal LTP current under normal conditions. When you add A beta in red to these neurons, you have a dramatic decrease in LTP. So your NMDA receptor is not functioning properly anymore. You can rescue that with an inhibitor against the NMDA receptor, and then it's going back up. Okay? So A beta apparently has a direct effect on the NMDA receptor. To be honest, I don't believe that. I mean, I believe this effect is pretty solid. Many people have shown that. But nobody has shown that A beta binds to the NMDA receptor. That is not known. But what is known is that A beta binds to LRP. So theoretically, and that's what we're doing, is now we want to find out whether the whole story I told you before could be basically mimicking this effect that A beta binds to LRP, then has an effect on the NMDA receptor, and we may be able to explain the story through that way. I don't know whether that is correct. It's just pure speculation at the moment. Okay. So that was the reason why I told you all the story about the TPA story before. So now we are back at LRP and A beta, and I would like to acknowledge the people who did this, and that was all former lab members, Anna Martin, Vladi Meyer, and Elaine Waldron. And Anna and Vladi, as the people I told you yesterday, went off to industry to earn money. And only Elaine, she is still in science. She is at the Johns Hopkins in Baltimore at the moment and doing well. So I'm the first one that stays in science. Excellent. And these are my collaborators. And tomorrow we will talk then more about NLP and its role in the blood-brain barrier, the role of the blood-brain barrier in general, and what that means for Alzheimer's disease. Okay, thanks a lot for your attention, and open for questions.
I'll turn that off.